Hello everyone, welcome to this interesting course, Introduction to Bioinformatics. Please read the course syllabus carefully. It contains useful information for you, such as the textbook that we are dealing with, or grading distribution, and also it includes the topics that we are going to cover during the semester. So let's start with lecture 1, which is related to the biological backgrounds that we need for this course. So first thing we should ask, what is bioinformatics itself? If I want to oversimplify it, we could say the goal of this bioinformatics is to analyze biological data using computer science or using computer simulations. But in order to define it more accurately, I need to remind you about some other topics because the definition varies from scientist to another one, and also to totally understand it, we need to know about other topics as well. So first thing we need to know is about molecular biology. And the data we are using in bioinformatics is related to molecular biology, not biology itself. So whenever we talk about data in bioinformatics, we don't care about, for example, ecology or other branch of biology. Everything is at molecular level. And statistics. A statistic is related to analyzing data, but not just about that. The process from collecting data and even reporting data is included inside the statistic, even summarizing your data. So we should know statistics in order to solve many problems in bioinformatics as well. And frequent question we receive from bioinformatics is that how related is that to biostatistics and how we can distinguish between them. As I mentioned, statistic is related to collecting your data, summarizing them, analyzing them, and reporting them. And whenever we are applying this to biological data, we call it biostatistics. So what's the difference between bioinformatics and biostatistics now? In biostatistics, we don't necessarily use computers in order to solve our problem. But in bioinformatics, we need computers in order to solve those problems. You see, there are just little differences. Sometimes they can be exactly the same. Whenever we are applying the statistical analysis inside bioinformatics, they look pretty same because in the statistics, sometimes we use computers, and not even sometimes, lots of times we use that. We have lots of softwares in order to deal with the statistics. So you see there are some intersection between them, but there are also some differences. And applied mathematics. When we want to define applied mathematics, we could say we want to formulate what's going on around us in this world, no matter what's the topic. It could be about a biological system, it could be about psychological system. Whenever we want to formulate it using mathematical modeling, then we call it applied mathematics. And computer science, you guys are already familiar with that. Whenever we want to, instead of using mathematical modeling, using computer simulations or using algorithms in order to describe and also analyze what's going on around us, then we call it computer science. Computer science is very useful because, as you know, many things we cannot calculate it manually, and we can use computers. So it could be another branch which is related to computational science. And as you can see, these sciences, they have lots of interceptions with one another. And this is the beauty of that. And bioinformatics itself is an interdisciplinary course, and it includes many sciences inside it. So now we can define bioinformatics much better, because it's not just as simple as analyzing biological data, because we need to use tools like computer science, applied mathematics, and the statistics in order to analyze those biological data. So to define it more accurately, we needed to define those terms as already mentioned. All right, but today we are going to focus more on biological context, because later we want to see how this is related to those sciences that I've just mentioned. Okay, central dogma of biology. It's related to how we produce protein from DNA. Let me use the laser here. Yes, we want to create protein from DNA. This process is central dogma of biology, and the way we reach phenotype from genotype. What do we mean by that? A genotype is considered as DNA level every information that we have inside our DNA. I will define them later in details. What do we mean by DNA? What do we mean by gene? You will see that. But just remember that genotype is related to our DNA level, and phenotype is related to our organism or tissue. And I will mention about the details later, that what do we mean, for example, when we say protein, what do we mean by tissue, very soon. And for example, when there's a mutation in our gene, it happens in the genotype process. And then later, when we have a disorder, this is related to phenotype. All right, so let's define DNA first. So DNA is a long chain of polynucleotides, and these nucleotides include these four, as we mentioned here, and we can just initialize them by just one letter, A, T, G, C. That's it. And as you can see for the PowerPoints, for example, we mentioned DNA, we give a short definition. Sometimes we don't even give it, but inside the first chapter of your book, they define them perfectly. So these PowerPoints are designed so that we can use them in the lecture, but it doesn't mean it's good enough for your exam. It means that you should go through the book for sure. Otherwise, it's impossible just to look at these slides and get the idea that what was DNA. So first we should read it, then why not? We can use it as a summary later. Okay, so as you can see, there are four letters here. And of course, in our body, they are not letters. This is the way, as computer scientists, we are thinking about them, but they are chemicals. They are basically chemicals. They are made of atoms. But the way we think about them, just we can consider them as the sequence of four letters. All right, let's see what is amino acid and how we can produce it. We talked about DNA, that we have sequence of four letters like A, T, G, T, C, T. Each three of them, we call them codon, and later, maybe on Wednesday, I can explain about intron and codon. But each three of them, when we consider them like this, they are responsible to create amino acid. So 3 by 3, they are creating different amino acids. And I will explain later how it happens. And the combination of these amino acids, they are creating protein. So you can also see the difference between protein and amino acid. So the combination of them are creating protein. Each one of these are amino acids. So if I change the place of this one with this one, then we will have a different protein. We will also talk about the protein structure later. And some codons like this, they are responsible for terminating this sequence. It means that there won't be other amino acids to be added to this one because we reached this sequence. And we will talk about the stop codons and the details later. So we need to know about these two terms. We use them a lot: transcription and translation. In order to produce protein from DNA, first we need mRNA or messenger RNA. What they are stand understand for what do we mean by that, please read first chapter of the book. There are so many details regarding them. Don't leave any words that we are using in the slide. If you don't know about any of them, please look up for them inside the book, or if a book is not good enough for you, then look it up on internet. But the book is well written. I'm sure you will find your answers there. All right, so transcription. What do we mean by that? It means that producing mRNA from DNA. In this process, we get one strand out of DNA, because as you saw before, each one of the letters were corresponding to a fixed number of letters. I will show you in the picture later that what do we mean by that. But the thing you should know is that when we say transcription, it means that producing mRNA from DNA. And how it will be done, for example, about translation and transcription, I will share with you some animation on YouTube, which are interesting. It could be useful. You can also look up for it. It's easy to find. And translation, it means that producing protein from mRNA. And for that, we need a ribosome. And again, you can look it up on in the book and also on internet. So this process is important, that we should from the beginning recognize what's happening here. So there are some kind of information in our DNA, right? There are sequence of these letters. As I mentioned, when I say letters, we are just looking at them like a computer scientist. And of course, there are chemicals. So these information are creating or conveying to mRNA. It's better to say also to transferring the information. Then mRNA, because DNA was located inside the nucleotide, right? It was inside the nucleotide of our cell. Then it will get out of the nucleotide, messenger RNA, and the information will be translated into amino acids, and the combination of these amino acids will create protein. We will again repeat and repeat this process, and I'm sure you will get used to it. From the beginning, if you don't remember the things that you learned from high school, maybe it gets tricky, but we will repeat it again and again, so it will be easy for you. So then proteins will create cells, and cells, which was the smallest organism that we know in our body, will create tissue or complicated organisms in our body, or even multi-proteins. And tissues together will create an organism. For example, human being is an organism. If we had different DNA, then we will be totally another entity. Maybe plants, maybe an animal. So you see, DNA could be very important in order to define who we are as an organism. And the collection of these organisms we call population of human being, for example. So again, let's go through more details related to transcription and translation. So DNA was double helix like this. For example, here we have T, we have A, we have C. Then T corresponds to a fixed letter, again A to a fixed letter. We know that. So if you know information about one strand, you should know about the other one. That's why usually they give you the sequence of one strand. It's good enough for us to predict what will happen. Now, look, when we are trying to transcribe DNA to mRNA, we divide these two strands from one another. You see 3', 5', it shows the direction. So whenever you see these two, it just shows the direction so that we don't mix them up. And for one strand, we show it like this. For the other, we show the other direction. So that if it goes from 3' to 5', we talk about, for example, this red line, and 5' to 3', we talk about this one. So in this case, oh, by the way, Here it shows in this picture that this is actually how we transcribe DNA to mRNA, and we have U here. We didn't have U inside the 4 letters that I've introduced to you. You see, A, T, G, C. And look at this one. We don't have any T, but we have U. You see, so there are just there's just one letter replacing, but the others will remain the same. A will be exactly A, C will be exactly C, and G will be C. G would be G, but T will be changed to U. This one, this picture actually a little misleading because it's not like T is transcribed to A. No. A will be always be A, and we are looking at it with different direction and a different gene. It's just for showing you that for mRNA we have now U instead of T. That's it. Okay, now after we transcribe our DNA, then we need to translate it into a protein. So how does it happen? We consider them 3 by 3. For example, AUG, this is a codon. And this one will be translated into this amino acid. And this is shortened by MET, but it has a full name. I will also show you. And in protein sequences, we usually show them with just 1 letter. We don't even show it with 3 letters. Again, I will show you later when we talk about databases, or maybe we can show it at the end of this session too. So 3 by 3, they are corresponding to different amino acids, as you can see. And for sure, it was a stop codon here so that we don't have anything after that. So the combination of them is creating finally this protein. So as a practice, if I want to transcribe, because I don't want you to be misled by this picture, let's say I want to transcribe this strand. T will be U, A will be A, C will be C, C will be C. So we will have an mRNA including U, A, C, C, A, C, A, C, U, C, G, if you consider this direction, 3' to 5'. As I said, you should read the chapter carefully at the beginning because you are computer scientists. It sounds new to you, especially if you forgot about your biological background from high school. But we repeat them again and again, so don't worry about it. But please read the first chapter carefully from the beginning. So we talked about cell, that everything regarding DNA that we talked about happens inside the nucleus of the cell. So it's nice that you guys go through this. It's not necessary for sure to memorize all the terms now. They can be useful, of course, if you know about them one by one, but we just show you that everything is happening inside our cell. DNA gets out by actually be transcribed to mRNA, and this is not like DNA gets out because the information is passing on outside the nucleotide, and that's why it's very important for you guys to watch those animations. So as we mentioned, we have DNA sequence. Like this is the way it is defined in our DNA, G, T, C, G, G, and so on. For example, in computer, we have binary sequence 0, 1, and you already know how to deal with that. But when DNA wants to transform or sometimes transfer the information, then it uses these 4 letters. And so it's different from computer by this. And that's why we need 2 bits in order to define it in our computers. This picture is very important in order to get a better picture of what we are talking about. So in our sequence, whenever we reach U, for example, This one will be translated into this amino acid. Or for example, GUU. This one will be translated into valine. So you see, some different codons, triplets like this, will create as the same amino acid. So this table is very useful for us to know that from which codon we can reach which amino acid. For example, if mutation happens in our cells in our gene by accident, then for example U changes to C, it's no problem because they will create the same amino acid. But if we have, for example, if U changes, then we will have totally different amino acid, and because of that, when the combination of them will create a protein, it will be totally different protein with different function. So it can be a big problem for our body, for our cell, and we should do research to see what happens in our body. Sometimes through phenotype, we understand what happened. What do I mean by phenotype again? For example, something happens as a disorder, then we understand, okay, this is a phenotype, and something bad happened in our body, then we should go through the genome level to understand what was the cause of that. And that's the genotype level that we were talking about. So you see, from these codons, we are creating amino acids. And instead of showing with 3 letters, as I shown in one of the picture, we show them by just 1 letter. It's easier when we consider them inside the sequence like this. So you see, instead of saying ALA, for example, for an amino acid, then we can use just a letter for that. And each letter, so here, corresponds to amino acids, and the combination of them is creating a protein, right? And we will talk about the details that how we can stop production, for example, from translation, and for example, to cut it from here and have a different protein. We will talk about those procedures later. So on Wednesday, I'm going to show you how to find these sequences, how to find protein structures, and so on. But for the time being, what I need you guys to do is to go through the lecture that we talked about today and get yourself familiar with biological context, because this is very important from the beginning. We cannot just neglect it and start reading it, for example, 2 weeks later. It's not possible. Let's do it this week, so please take it seriously from now. Start chapter 1. And don't worry about it. This course is very interesting and very easy, but it needs just studying. If you study, if you do it week by week, there won't be no problem, and it's going to be very useful for you, especially if you want to continue your education as Master of Science. So if you have any questions, please let me know. Otherwise, we will continue this lecture on Wednesday.